All right. Thanks everyone again for joining us today. My name is Cory Nelson. I work in the College of Sciences here at UNLV, and we're really excited to have you joining us for our June session of Virtual Lab Notes. Today we have a presentation from Dr. Cory Rusinek in the Department of Chemistry and Biochemistry. But just a few quick things before we get going. Make sure that your microphone is muted and stays muted throughout the entire presentation, and that your video also stays off. It helps to preserve bandwidth throughout the presentation. Also, you can use the chat function if you're having any problems or issues, or if you have questions. We'll ask questions at the end of the presentation of Dr. Rusinek. So if he says something really interesting, hopefully he says a lot of really interesting things, but if he says something that you want to know more about, feel free to pop that in the chat and we'll ask those questions. If you have any issues with Google Meet throughout the presentation, just make sure that you visit the Google Meet support link. It's support.google.com/meet. You can try to message me as well. I'm not super technologically savvy, so I will do my best to help, but we've so far, fingers crossed, haven't had any major issues using this platform. And then just as a reminder, we are recording this presentation and it will be available on the College of Sciences website as well as the UNLV College of Sciences YouTube page. So if you missed something, feel free to check it out afterwards. It usually takes us about a week or so to get that up there. And with that being said, I'm going to introduce next Virtual Lab Notes topic, which will be in August. We're going to take a little break in July. A lot of people are on vacation, so we're going to move to the next month of August. And we'll be hearing from Dr. Laurel Raftery. She's a professor in the School of Life Sciences. She's a cell biologist and does some phenomenal work and research on genetics and cell mutations. And so we'll have Dr. Raftery joining us on August 25th. So mark that on your calendars. And with that being said, I will introduce this month's speaker, which is Dr. Cory Rusinek. And Dr. Rusinek is an electroanalytical chemist and an assistant professor in the Department of Chemistry and Biochemistry at UNLV. He is also a faculty member in the UNLV Radiochemistry PhD program. He joined UNLV in mid-2019. Dr. Rusinek received his undergraduate degree in chemistry from Case Western Reserve University before completing his PhD in analytical chemistry at the University of Cincinnati under the advisement of Professor William R. Heinemann. Dr. Rusinek's research is driven by biomedical, environmental, and nuclear energy-based applications. Currently, Dr. Rusinek's research is supported by funding from the Department of Energy, Nuclear Energy University Program, and the Nevada IDEA Network of Biomedical Research Excellence. Hopefully I said that right, Cory. And in the DOE Minority Serving Institution Partnership Program. Dr. Rusinek is a native of Northeast Ohio near Cleveland, and we're very excited to have him with us today. And without further ado, I will turn it over to you.
All righty. Thank you so much, Cory, for the nice introduction. And thank you to everyone for attending today. I hope that everybody's able to take something away from this presentation, and certainly a greater appreciation for the field of electrochemistry at large. So let me share my screen. Okay. All right. Okay. Everybody see this okay? I assume so.
We can see it. It's not in presentation mode, but we can see it. There you go. Perfect.
All right, cool. All right, again, so my name's Cory Rusinek, and I am an assistant professor in the chemistry department and as well as the radiochemistry program. And my talk is titled Electrochemistry is Everywhere. So what is electrochemistry? So it's really the studies at the crossroads of electricity and chemical change. So we're going to go into that in quite a bit of detail today. And really what we're going to be looking at is the electrode surface. And I perhaps don't emphasize this with the images and in the text throughout the presentation, but I'll emphasize it right now that all of the reactions that are studied via electrochemistry, and I guess I'm speaking more from an electroanalytical perspective since I'm an analytical chemist, but all the reactions that we study are happening at the electrode surface. So the electrode material, while there's many factors that play into the performance of an electrochemical cell, you can really only go as far as your electrodes can take you. And that's one thing that I'll highlight later in the presentation, but I want to make sure we hammer that home now, is that these reactions are occurring at the surface. And essentially, for most of the cells we're going to be talking about, we're going to be applying an energy to that electrochemical cell or through that electrode surface, and that's going to cause this chemical change to occur. Now, sometimes energy isn't needed and the reaction is spontaneous, and that's what we're going to go over first with respect to batteries, and especially lithium-ion batteries, where when the battery's discharging, we have a spontaneous reaction occurring that we don't have to input energy to. So again, at the end of this presentation, hopefully you'll have a appreciation and importance for electrochemical cells in everyday life, and hopefully everybody leaves this presentation wanting to go home and become an electrochemist if you haven't chosen your career path already, or if you have, maybe you're going to want to go back to grad school and become an electrochemist. Anyhow, so before we jump into some fundamentals, let's talk about some applications, or general applications rather. So I talked about batteries already, so I'll highlight that first. So here we got a Tesla car up here, powered by a myriad of lithium-ion battery cells. We have an iPhone down here that is powered by a lithium-ion battery cell, and we have some disposable alkaline batteries down here, all of which are an application of electrochemistry that I'm sure everybody has seen in their everyday life. Here is something completely different. This is a system to do wastewater treatment, where you can flow water past a series of electrodes that's contaminated with some sort of organic toxin, and you can actually blast that organic toxin into CO2 and water and remediate it from a solution. We'll talk about that. Here are a couple of sensors. So right down here is a pH meter and a pH sensor. So if anybody has ever used a pH meter and a pH probe in the past, you have already done an electrochemical measurement. And here's what we call a 3-in-1 electrode sensor. We'll talk about this a little bit later. And these molecules here are istatin, which is a biomolecule associated, or a biomarker for stress, essentially. But you can also rig an electrochemical cell as a little sensor like this to measure some sort of analyte in a water sample, biological sample, what have you. Here's some solar panels. Again, I after making the presentation, I didn't really get a chance to talk a lot about solar, but I'm happy to, if somebody's interested in learning more about electrochemical applications of solar outside of this, I'm more than happy to chat with you. And then lastly, sort of an application that is maybe indirect and something you might not consider is, I'm showing this credit card here, and credit cards by and large are made by PVC. And we're going to talk about an electrochemical cell that is used to generate chlorine gas, which can and is used to generate PVC. So I'm sure everybody's heard of PVC piping, has heard of PVC in general as a household item as well. And there is a part that an electrochemical cell plays in the formation and development of PVC. So again, before going into all these slides, I figured it'd be a good idea to go over some terminology. Again, I know this presentation is for a general audience, so I tried to keep things fairly general. And this is going to be sort of a survey of electrochemistry as a field, including analytical chemistry as well. But the basic terminology is pretty much the same across electrochemistry in general. So we have the electron, that's kind of our star of the show here, and that's the reactant. So we're going to look at a variety of different scenarios where electrons are transferred, and whether that's a battery, whether that's a sensor, what have you, that electron transfer is what's either giving us energy or is what we're using to sense current or measure current and correlate that to a concentration of something in solution. So the electron is really our main reactant in all of these studies or in applications that I'm going to show. The electrode, I sort of already talked about the electrode. This is where the reaction is occurring. And again, you can really only go as far as your electrodes are capable of going. A little bit more in detail, we got oxidation and reduction. Oxidation is the loss of electrons, increase in oxidation state, but really just consider oxidation as the loss of electrons. Reduction, gain of electrons. Anode and cathode, these are two types of electrodes. In general, the anode is where oxidation will occur, and the cathode is where reduction will occur. And two, again, very general terms, current and potential. So current is essentially the flow of electrons. And potential is what I want you to think about as this expression of electron energy. So I'm going to show you some equations in a couple of slides that relate the potential to energy. And I want you to think about this potential scale, and I'll mention voltage and potential and what have you. And that's really an expression of energy when you really talk about it in an electrochemical sense. And here's some silly, again, this is a talk given by a chemistry faculty, so I'd be remiss if I didn't include some dry jokes here and there. Okay, so an electrochemical cell at large. So that's what's shown over here. This may be one of the more technical slides in the presentation, so bear with me here. But what you need to form an electrochemical cell is two half cells. And those two half cells, as shown over here on the left, anode, over here on the right as cathode, these have their own makeup and constituents that are separate from one another, but they are still ionically connected as well, in conjunction with being electrically connected through the top here. So a little bit more detail. What do we have in our anode compartment? We have a piece of zinc, so we'll call it our zinc anode. And over here in the cathode, we have a copper electrode. In each of those solutions, there is a salt or a solution that contains those ions. So in the case of the zinc anode, we have a solution of zinc sulfate here. And over here at the cathode, we have a solution of copper sulfate. And both of these electrodes are electrically connected or hooked up in this sense to what says a digital voltmeter, but they can be directly electrically connected to one another. Now, this little guy here is called the salt bridge, and there's no electricity per se or electrons flowing through the salt bridge here. But what the salt bridge allows is for the passage of certain ions back and forth. So while it's crucial, obviously, to have both of our electrochemical half cells electrically connected to one another so that we can either apply energy to make a reaction occur or hook them up and allow a spontaneous reaction to occur where we're going to get energy from, we also have to have equal ionic flow between those two half cells. And that's what this salt bridge does here. As you can see, it's saturated with potassium chloride. So over here in the cathode compartment, we have this interaction at the tip of the salt bridge between potassium and the sulfate from the solution. And over here, we have the same thing occurring, but with zinc from the solution and chloride from the salt bridge. So while the electrons, depending on how this is electrically connected, are flowing through the electrodes and into the electric circuit and then back into the other electrode, these ions that are in each solution are going to be passing through the tips of the salt bridge to make sure that the two half cells are ionically connected to one another. So in the case of zinc and copper, we're going to show this on the next slide, but we have these characteristic reactions occurring. So in our anode, we have the zinc electrode oxidizing to zinc 2+, with 2 electrons going into the electric circuit rather. And then in our cathode, we have copper ions depositing on the electrode as copper 0. So here are our two reactions here. The electrons have to balance out one another, and we have our overall cell reaction down here at the bottom. And again, we're going to talk about more of this on the next slide. But if there's anything I'd like you to take away from this slide, it's that you have to have two separate half cells that are both ionically and electrically connected to one another for your electrochemical cell to function, whether that's a cell you need to apply energy to cause a reaction to occur, or if it's a cell that you're setting up electrically connecting, and then it's a spontaneous reaction occurs and gives you some sort of energy output. And I have this little pictograph here. It says, when life gives you lemons, construct a crude electrochemical battery. Well, I don't know if anybody has seen that before, but you can hook up some electrical leads into a lemon, and you can have it power a light bulb, because the lemon itself acts as this salt bridge, and all of the citric acid, or the high concentration of citric acid in that lemon, allows you to have that one ionic conductance. It still separates the two half cells from one another. So you can have electrochemical reactions occurring on two sides of a lemon. You're essentially polarizing the lemon with a positive on one end and a negative on the other. And you can string up a whole bunch of lemons in sequence with one another to give yourself even more power output, or light your bulb lighter or for a longer period of time. Okay. So again, these are the more technical slides in the presentation, and I don't want you guys to get caught up in all these equations here, but I just want you to look at this one up here, where this shows a reaction scheme that is just like the one that we showed over here with zinc and copper. And so we have A plus some number of electrons goes to C, B goes from oxidizes to some number of electrons to D. And our overall reaction is shown right here. A plus B goes to C plus D, and the electrons are always canceling themselves out. So now I want you to look right here. So this delta G cell, or delta G, is the change or the Gibbs free energy. And what does that mean? It's just a fancy way to say basically an energetic driving force for a reaction to occur. So the Gibbs free energy will tell you whether or not basically how much energy you need for a reaction to occur, or how much energy you might get from a spontaneous reaction. And you want delta G to be negative for a spontaneous reaction to occur. So when we have a negative value for delta G, our reaction is going to be spontaneous, and therefore we're going to get energy out of that system. Now you can relate that free energy to the cell potential of an electrochemical cell through this reaction right here. And this shows minus N, which is the number of electrons transferred. F is a constant, Faraday's constant, and then times your electrochemical cell potential. So when you look at this, again, you don't have to really think about it too hard, but you can directly see that when we have a positive value for the electrochemical cell potential, we're going to have a negative value for delta G. So if we have an electrochemical cell where we design and the cell potential turns out to be positive, more likely, or not more likely, that reaction is going to be spontaneous, because it's going to correspond with a negative value of negative free energy value per this equation here. Now, these two equations right here, this is two forms of the Nernst equation, which are the two most fundamental, or the most fundamental and essential equation in electrochemistry, but we're not really going to go into those because I don't think that it's particularly necessary. But for this first part of the presentation after this part, we're going to talk about these two reactions here, where we have our cell potential, which is E cell, that's our overall cell potential, and that's equal to the potential of the cathode compartment minus the potential of the anode compartment. So we can see that here, our overall cell potential is going to be the potential of our copper half cell minus that of our anode compartment, which is our zinc half cell. So here's where I'm going to write for just a second. Let's see here. 1 second. There we go. All right. Can you still see okay, Cory?
Yep.
Okay, cool. So in this part now, we're going to look at a couple of these different reactions. So that representation I just showed you, we had a zinc anode and we had a copper cathode. And this table over here is called a standard reduction potential table. And you can see there are a whole bunch of different reactions that are shown here, and the electrochemical potential at where those will occur over here. And again, when I said the potential as an expression of energy, you can think of this over here on the right almost as an energy scale. So we come down here, and I should have also said there are a lot more reactions than just what's shown at this table. So let's just think about what we had in our cell from a couple of slides ago. So we had a cathode compartment. That was the copper 2 plus 2 electrons goes to copper 0. And we see that that has a potential of positive 0.34 volts. Okay. And then in our anode, we had zinc, which is down here. We're going to write that in the opposite direction, but it does not mean that the sign of the potential is going to change. So we have zinc 0 goes to zinc 2 plus 2 electrons, right? And so our overall reaction, or I'm sorry, this is at minus 0.76 volts. So our overall reaction is going to be the copper 2 plus the zinc 0 goes to copper 0 and zinc 2 plus, and the electrons are going to cancel out. But that equation that I just showed on the previous slide for E cell equals E cathode minus E anode, we're going to have positive 0.34 volts for our cathode compartment. That was the copper reaction. And then we have minus a negative 0.76 volts for our zinc reaction. So now if we just do that simple math, right, E cell equals positive 1.1 volts or thereabouts. So if E cell is positive, remember that free energy reaction. Okay, E cell is positive, so therefore delta G is going to be negative. So therefore this reaction would be spontaneous. So that means if we were to go back and make that cell from a couple slides ago and just directly connect those two leads, the lead from the copper electrode to the lead from the zinc electrode, that reaction would proceed. We would get 1.1 volts out of it until the reaction went to completion. So that would be a way that we could get energy out of an electrochemical cell. So with that in mind now, first thing we're going to talk about from an application standpoint is the lithium-ion battery. And I want to preface what I'm about to show you with the fact that the electrochemical cell for a lithium-ion battery is drastically more complex than how I'm going to describe it. But if you just look down here at lithium, it's right here. It's the most negative value on the table, minus 3.04 volts. So let's say we were able to just sub the lithium in for the zinc, right? So our anode would then become the lithium 0 goes to lithium plus, whoops. Sorry about that. Goes to lithium plus an electron, and that's at minus 3.04 volts. If we keep, and we're keeping the copper cathode the same. So now if you see, if we sub the lithium in for the zinc, we now have a overall cell potential of positive 0.34 volts plus 3.04 volts. And now our cell potential is what? 3.38 volts positive. Okay, so even more. So now we're going to get more energy out of that system by having the lithium anode there. So there's one of the first reason why you can see a lithium ion battery might be more powerful, quote unquote, than a, let's say, traditional alkaline battery or a zinc containing battery. Okay, and I'm going to reattach here. All right. Sorry for my chicken scratch handwriting, but hopefully everybody was able to take away from that the the electricity, just swapping out the electrode and the half cell there, we're able to get quite a bit more energy out of that. So now if you think about when we have to recharge that lithium ion battery, that's when the signs are going to change and this value is going to become negative. The E cell value would be negative. So what can you say? What does that mean? Well, a lithium ion battery doesn't recharge itself. We have to plug it in and recharge it. And so I think for the sake of time, I'll go through this slide relatively quickly. But essentially a galvanic cell is one that when you hook everything up, you get an output of power from it. So in this case, we have a light bulb. And over here, an electrolytic cell is when we plug that cell in and we drive it in the opposite direction. So this is essentially how a rechargeable battery is going to work. Okay, so that's kind of the background of the more electrochemical theory, rather. So now we can start talking about some different applications. And it's likely we're not going to make it through all of these depending on time, but I have a handful selected if we do start to run out of time. But first, we're going to go over some industrial and commercial electrochemical applications before getting into analytical applications here towards the end of the presentation. So the lithium ion battery looks pretty similar to what we just showed. We have an anode compartment, we have a cathode compartment. We don't have a salt bridge, but we have a separator that allows for the lithium ions to diffuse through or across. So typically, I call this the positive electrode over here. In a lithium ion battery, this positive electrode is typically going to be what contains the lithium. So I have written here lithium cobalt oxide, lithium iron phosphate are two very common lithium ion batteries you'll see on the next slide with electric cars. There's a couple other alloys that are used. And the negative electrode here is, I denoted as graphite, but there's plenty of other materials that have been used like graphene, other carbon materials. And there's been quite a bit of research with respect to developing different types of carbon electrodes for, or to use as the anode. But essentially, we'll talk about the separator and the solid electrode interface here in a second. But when you're charging a lithium ion battery, the lithium ions are essentially leaving your lithium alloy, passing through the separator and depositing onto your carbon-containing electrode over here. And in order for that to happen, we have to input some energy. So that's when we're charging the battery. So when you have your cell phone in your hand, you're run out of battery, you need to recharge it. So this is what's happening. You're plugging your phone into the wall, and the lithium ions in your battery are traveling from the lithium-containing cathode to the carbon-containing anode, as it's denoted in this figure. And then once your phone is sufficiently charged, that basically means there's no more lithium transporting from the lithium cobalt oxide, in this case, to the carbonaceous material over here. Then you could say your battery's charged and it's ready to use. So then you unplug it from the wall and you go about your business using your phone. And all the while that you're using it, the lithium is now transporting away or off of the carbon-containing anode and back to this lithium cobalt oxide containing cathode over here. And so we have these lithium ions essentially moving back and forth. But as I said with the original electrochemical cell, we have to have ionic flow as well as electrical flow. So the lithium here is what's providing our ionic flow, and it's the electrons flowing in either direction, depending whether it's being charged or discharged, that are giving us our electrical flow. And in the case of a discharge, this is where we're actually harvesting our energy from. Okay, so when a lithium-ion battery is discharging, it's a galvanic cell because we're getting we're getting energy out of it by a spontaneous reaction occurring. And when it's charging, it's an electrolytic cell because we are driving the reaction in the opposite direction. So one little note on this separator and solid electrolyte interface, you might know that as you charge or as you use a lithium-ion battery, you lose some capacity over time. So your phone might not last, or your computer might not last as long over time. And that's because some of the lithium can get caught over time in this solid electrolyte interface. And if you remove some of the lithium that's capable to move back and forth across this interface from one electrode to the other, you have less lithium there, and you're just overall going to reduce the battery capacity. So that's why you'll hear sometimes don't charge your phone with your iPhone with a computer charger, because the voltage and current that's flowing into the electrode from the outlet is going to be quite a bit higher with a computer charger than with your phone charger. Yeah, your phone might charge a heck of a lot faster, but you're moving this lithium a lot faster across this interface, and the likelihood that some lithium gets caught in this solid electrolyte interface to never be recovered is much higher, and therefore you'll decrease the performance of a battery quicker if you charge it with a charger that's much higher rated than your battery is rated for. And the same for the same reason where you might not want to discharge your battery super duper fast, and you might lose some lithium in the solid electrolyte interface. So that's lithium-ion batteries in general. Again, I'm not a battery chemist, and again, I'm an analytical chemist, but the theme of this presentation is to show you how diverse electrochemistry is and all these different applications of it in general. So that's why I wanted to cover that. I am going to be way short on time. Anyway, so the 2019 Nobel Prize in Chemistry was actually awarded to these three fellows here for their development of the lithium-ion battery. So this is a multi-billion-dollar industry now that is only growing. and again, a hallmark application of electrochemistry and its impact on not only today, but the future as well. So how does a lithium-ion battery power a Tesla? As I said, the electric cars use a slightly different lithium alloy containing electrode, where Tesla uses this lithium nickel cobalt aluminum alloy. Most other companies that produce EVs use a slightly different alloy. But really, it's not just one gigantic battery that is powering a Tesla. It is thousands of small lithium-ion cells. So here is a Tesla Model S battery pack here. And here's kind of a little schematic that shows kind of a ghost image of the car around it. But it has thousands of these lithium-ion cells rather than just one. And this has several economic and safety advantages. From an economic perspective, let's say you have, just a handful or maybe even just one of the lithium-ion cells that fouls or goes bad or what have you, you can replace just maybe that one or several cells and salvage the rest of the battery pack so that it can be continually used. So there's some economic benefit there. And then there's also this benefit of I don't want to call it heat dissipation, but there's just like a less likelihood of the battery overheating when you have thousands of small lithium-ion cells rather than one very large lithium-ion battery. So that, yeah, it's a common, I didn't know that until I had looked into this and started learning about Tesla, that there were just thousands of small lithium-ion cells that comprise the car and the battery itself. But that's how they work. And the chemistry is, again, slightly different. Again, all these electric vehicle companies are, these are patented, and they're not going to tell you everything about their process and their electrodes. That's the whole part of trade secrets. But in general, that's the process for how these work. So to wrap that up, here's the Tesla being charged, batteries being charged. It's an electrolytic cell, okay, charging the battery. And then here it is cruising along somewhere, probably in California, using the battery, and is now a galvanic cell. So you're basically just driving around a big electrochemical cell on wheels when you're driving a Tesla. Okay, so switching gears a little bit, and I think I'm going to have to go a little bit faster through these parts, but because I've only got about 15 more minutes. And but the chlor-alkali process is a similar cell design, but for a completely different application. And what the chlor-alkali process is called the electrolysis of brine, and it's an efficient way to produce chlorine gas, hydrogen gas, and sodium hydroxide, all of which have their certain uses. So if you look at the cell here, on the left side, we have 26% sodium chloride coming in, interacting with an anode, being oxidized to chlorine gas. That's what's shown in this reaction here, and it's leaving a slightly less concentrated sodium chloride. Here we have a separator that allows for sodium ions to pass back and forth. So we have our ionic conductance between the two half cells. And over here we have our negative electrode, where water is being pumped in, being electrolyzed by the negative electrode, and forming hydrogen gas and hydroxyl ions. Those hydroxyl ions pair up with the sodium ions that are diffusing across the separator from the anode compartment, and they're forming sodium hydroxide. So we put sodium chloride and water in, and we get out chlorine, hydrogen, sodium hydroxide, slightly less salty water. And here's our overall reaction here. So as this is occurring, for every 1 mole of chlorine and hydrogen gas, we get 2 moles of sodium hydroxide. So you have to have some good use for all 3 of these things to make this process really useful, because it does take a lot of energy for this to happen. Because again, this is an electrolytic cell. We can't just pump these in and have these 2 electrodes in there and expect these reactions to occur with this cell. This is an electrolytic cell where an external energy source is being applied through these electrodes to cause these reactions to occur and give us these useful products. So in the effort of time, again, I mentioned already, chlorine gas is used to make PVC. There are certain chlor-alkali process cells that are used to chlorinate swimming pools, which is kind of cool. So you don't have to use chlorine tablets. You have a little electrochemical cell making your chlorine. And tens of millions of tons of chlorine gas are prepared by this process every year. And with respect to sodium hydroxide and hydrogen gas, hydrogen gas can be used to make these few chemicals here. And sodium hydroxide is essential to the chemical industry. And I think that the chemical industry in general gets overlooked from that perspective, where, you might look at your household cleaning supplies or your shampoo or whatnot. You see all of these names, these chemicals you can't pronounce. But by and large, there's a lot more simple chemistry that occurs and simple chemicals that are used to synthesize those. And a lot of things like sodium hydroxide, acids, ammonia, hydrogen peroxide can play a role on a lot of those things. So you might not see that as tangible evidence of the usefulness of this process, but just know that these chemicals that are listed here are essential to the chemical industry, which is a much larger industry than I think is than thought of in everyday life. Okay. I think I'm going to have to skip through wastewater treatment to save on time. But what I think what I can say is that in a similar cell design as what we saw previously, the chlor-alkali process and a battery cell, we have an anode and a cathode, but we don't have to have the separator here. And when we can take these electrodes, put several of them next to one another in sequence, so anode, cathode, and we can apply a very high potential between the anode and cathode. We could have 5 total electrodes. We might have 2 anodes and 3 cathodes. And we can apply tens of volts and several hundred milliamps or high current densities between those electrodes, and it'll give the anode enough energy to destroy an organic contaminant in solution. So we can stick these electrodes in water, apply this very high potential on current through the anode surface here, and we'll flow let's say wastewater past the electrode surface, and the contaminant in that wastewater will interact with the anode surface. And because it's at such high potential and current, we can actually break down an organic contaminant into its constituents. And that's what's shown here, where we have the organics interacting with the electrode surface and being taken to carbon dioxide. In this case, that's some nitrogen gas, hydrogen gas, and essentially just water. So if this is something that you're particularly interested in, please feel free to reach out to me, and I'd be happy to discuss some more. But there's a wide variety of contaminants and emerging contaminants in drinking water and wastewater that are very recalcitrant and difficult to remediate by conventional remediation techniques. Let's say like activated carbon and like what's in your Brita filter. That typically is not enough to remediate something like a per and polyfluoroalkyl substance, which can bioaccumulate in your body if you're drinking it in your drinking water for long periods of time and cause a whole slew of health issues. So electrochemistry has some roles to play in destroying contaminants in a wastewater matrix. I think I'll skip ahead to here and show here's one specific treatment train for PFAS. So again, these perfluorinated or per and polyfluoroalkyl substances are very difficult to remediate and cause a lot of health problems because if you ingest it in your body, they just stay there. They don't leave your body. So what this is showing is using an ion exchange resin, which has been shown to remediate PFAS from contaminated groundwater and wastewater. But over time, kind of like if anybody has a water softener is basically extracting, if you will, the ions in water like magnesium and calcium that make your water hard. And then over time, your water softener has to regenerate itself because it becomes saturated with calcium and magnesium. It's kind of the same thing here, except rather than it being a water softener, we have PFAS contaminated water going through a very specialized ion exchange resin. And then that resin is very expensive, so it needs to be regenerated the same way your water softener needs to be regenerated. And with a water softener, the concentrated magnesium calcium solution just goes right down your drain because those aren't really considered contaminants, magnesium and calcium. But with PFAS, you can't just dump it down the drain because you'd be just perpetuating the same issue over and over again. So what the electrochemical technique can be used to, after that resin has been regenerated, rather than just dumping it down the drain, well, let's take that concentrated regenerate solution and flow it past some electrodes and blast all those PFAS to carbon dioxide, water, and fluoride ions. Whoops. There we go. So again, this is a useful way as a destructive technology for an emerging contaminant like PFAS. And the reason you can't just say, oh, well, I'm going to go just plug in an electrochemical cell at a wastewater treatment plant and just flow all the water past the electrodes and destroy the PFAS. Well, you have to also consider that these are electrolytic cells that are doing this work. And so we have to input the energy. And when you're applying tens of volts and hundreds of milliamps per centimeter squared through an anode surface, when you think of that at an appreciable scale, the energy costs are just astronomical. So electrochemical oxidation has found a nice little home to treat these small volume, highly concentrated regenerate solutions, but still a part of the overall treatment train with, again, a very similar cell design. We don't have the separator here because we don't technically need it, and I'm not going to go into why, but essentially we still just have an anode and cathode here, and we're just doing something different. Okay. Now for the last 10 minutes or so, I think I'll go through analytical electrochemistry and what we do at our group at UNLV. So in general, electroanalytical chemistry is the study of analyte concentration, redox properties, chemical properties, etc. A number of different properties that can be used, but primarily what we're going to talk about here is quantifying a concentration of a species in solution. So we have a slightly different electrochemical cell design to do that, and we have precise control over the potential. And by and large, we'll be controlling that potential and measuring the current output that we get from an electrochemical reaction. And by and large, that a magnitude of that current is going to be proportional to concentration. And what's the big advantage here is that electroanalytical chemistry is cheap, easy to use, and easy to miniaturize. So we're a potentiostat, which is the instrument that we need to do electrochemical measurements, and our consumable electrodes might be for you can get a great potentiostat for $5,000, $10,000. Some advanced systems I put here can be in the $50,000 range, but competing technology is typically 10 to 100 times more expensive. It might be a little more sensitive and maybe a little more selective. So electroanalytical measurements and sensors aren't universally accepted and usable, but there are a number of different applications where that ease of use, portability, all these things really are hallmark advantages that you can use. And over here, I'm going to talk about a glucometer, but this is a glucometer here to measure glucose in blood. And most of those are electrochemical sensors. There's just a reaction between the glucose and a glucose oxidase enzyme that's coated on the electrode surface. There is a current response associated with that reaction between the glucose and glucose oxidase, and the current that is produced is measured and correlated to concentration. So there's a quick and easy little electrochemical measurement that I'm sure everybody is familiar with in doing glucose monitoring in blood. But now for this part of the presentation, we have to consider a slightly different cell design, where before we only had 2 electrodes, but now I talked about how we have to have this very precise control over the potential. We want to be able to control it down ideally to the microvolts, so 10 to the minus 6 volts. So we want to have very precise control over that, and we want to be able to measure current responses that can be as low as nanoamps, which is 10 to the minus 9 amperes, so very, very small currents. So to do that, we add a third electrode, and that's called the reference electrode. So this says working reference counter. I just want you to not worry about the jargon here and just consider working and counter electrodes as our anode and cathode, rather. And the reference electrode is just kind of getting stuck in there so that the potentiostat knows what its zero is. So if we tell the working and counter electrodes, our anode and cathode, to go to positive 1 volt, well, the instrument doesn't know what that is, right? That's like saying, how far away is Los Angeles? And you say, well, how far away is Los Angeles from where? From Vegas, from Phoenix, from Denver. You have to have a point of reference so that you can say, okay, I'm going to start here and I'm going to get to LA in 4 hours. You start here and you get to LA in 6 hours. It's kind of that same sort of concept here, where the reference electrode is your point 0 for you to take your other electrodes to that precise potential that you want, so you can have a redox reaction occur and measure a current response out of it. And I'm just going to go through a few applications here. Here is a device that is used and taken out into the field. Here's our 3 electrodes here. It looks like they have an additional, probably a gas line here. But this is called the metallizer, and this is very common. It's sold by a company in the UK. and you can essentially take this out in the field. You can scoop up some water, add one other reagent that they give you, stick the electrodes in there, and it can do measurements of arsenic. It can do measurements of lead, zinc, calcium, of a wide variety of inorganic ions can be measured with this. And again, this is something that just folds up into a case. You can take it right out into the field. And this is something that you typically can't do with other analytical instrumentation that can get down to a parts per billion, parts per million detection limit. So that's an environmental application. Here's a biological application that does the exact same thing. This device is called the LeadCare 2. Here's our electrode device over here, slightly different, but we still have a working electrode, counter electrode, and a reference electrode. And essentially what we do is we plug that right into the system here. You take a prick of blood off of somebody's, toe or finger, mix it with their secret solution, which is dilute hydrochloric acid. And then that solution is just placed right on the area where the electrodes are containing. You plug that in, press play, and it does a electrochemical measurement, and it gives you a response that is going to be proportional to concentration. And for this specifically, again, this is for LeadCare, or to detect lead, rather, I'm sorry. And you can find these at a lot of hospital laboratories and whatnot to do rapid detection of lead in blood, which lead has become a big issue in many areas around the country. And the advantage is that this uses such a small sample size. So this can be used for infants as well to measure the blood lead levels in children. And that's a yearly checkup test that is done in a lot of areas now amongst children. So what do we do at UNLV in this area? So we are developing a green chemistry extraction method to improve detection of lead in water. The limit for lead in water is 15 parts per billion. So extracting and pre-concentrating could make detection much easier. So if you think of 15 parts lead per 1 billion parts water. So that's the action limits, or when the EPA has to go do something about drinking water, is when it's above that level. And that's a very, very low level if you think about 15 parts lead per 1 billion part of solution or water. And so what we're trying to do is to develop a method where we can take lead, let's say at 10 parts per billion, 100 mils, and we can pre-concentrate that down by 100 and now it's 1,000 parts per billion in a 1 mil solution. So we significantly pre-concentrate and we do our analysis in the extracted media. And that's kind of what's depicted over here. We use a surfactant and a few other chemicals in order to make that happen. And just to show the advantages here, the CPE is the extraction method. I wasn't going to go into in great detail, but without the extraction, we can't really see lead at all. We do the extraction, we get this very nice response that's characteristic of lead electrochemical reaction. And as you can see here, we have current on the Y and potential on the X. So we're precisely controlling the potential because we have those 3 electrodes now, and we're monitoring the current at these different potentials. And we know that lead, the reaction is going to occur in this potential range. So we'll take the magnitude of the current we get, and that'll be proportional to concentration, and therefore we can build a sensor out of that. Let's see. All right, so I think I am running out of time. I think I only got about 5 minutes left. I don't have that many slides, but
there's not that many questions, Cory, so you can go for a little bit longer. Yeah.
Cool. All right, awesome. So here is another really cool way to do an electrochemical measurement, and that's to do it in the brain and in the body. And I show Frankenstein down here because Frankenstein was made by sticking some electrodes in his brain to stimulate it and what have you. And by and large, that's entirely possible. Stimulating the brain with electrodes isn't necessarily pure electrochemistry, but it is done. But what I'm going to talk about is actually quantifying neurotransmitters in the brain. And so in the 50s and into the 60s is when it was found that dopamine behaves as a neurotransmitter in the brain. Dopamine makes you feel good. And around in the mid 60s, that was when the first studies were done by implanting carbon electrodes in a mouse or a rat brain and being able to measure dopamine in real time. So this is one of the first rigs of trying to measure a dopamine response from a rat. And there's some little microelectrodes we'll show in the next slide. And this field has blossomed into a whole slew of different studies and addiction studies, understanding depression and what have you, because all of these neurotransmitters and catecholamines, for that matter, are electroactive. So they'll undergo electrochemical reactions and you can study them. And so that's what's shown here. So let's look up at this top left figure here first. This is the size of the electrodes. These are microelectrodes. Everything else I've shown before has all been something that's either been about the size of your hand, let's call it. But now we're talking about an electrode that's the fraction of the width of your hair. So here's the tip, about 5 micrometers in diameter. Your hair is about 70 micrometers. So it's quite a bit smaller. And that is taken and implanted into a rat brain. And there's also a reference electrode in there as well. And so what you can do is you can, or a lot of studies have done, is they'll put this rat under some conditions that will cause neuron firing and release of dopamine. So one study I read about was they actually were administering, had the electrodes in the rat brain. They also had a way that they could feed in a drug of some sort to it. And the rat was in the cage with the electrodes in its head, and it was going up and it would touch this button. And every time it touched the button, cocaine would get administered directly to the brain of this rat. So, they did this for some number of time. It would touch the button. It would get the rush of dopamine that's characteristic with drug seeking. And then it would have this influx of cocaine in the brain. So they started to do some more studies where then the rat would after it got used to touching the button and getting this feedback mechanism, they started doing it where the rat would touch the button and it wouldn't get the cocaine administration. And they found that there was actually just as much dopamine released with the buildup to that rat going over to touch the button as there was when cocaine was actually administered. So they found that, again, the levels of dopamine associated with seeking the drug are almost gives you just as much of a high, rather, as actually doing the drug itself. And that was a pretty interesting breakthrough with respect to certain behavioral science. And again, a really cool application of electric. Again, it hasn't been done in humans; it's been done in animals. But nonetheless, it's really interesting. And sake of time, I won't describe this over here. But again, here's what our output response will look like. So it looks a little different than what I showed before. But here's our dopamine oxidation peak. So a real nice peak associated with our voltage on the X. So we're precisely controlling that still. And here's our current on the Y, where we can relate that back to concentration. So, really, really interesting technique. And the last thing I'll mention about is that this voltage range is being scanned so that this reaction will occur. And this is being scanned at the millisecond time scale. So when that electrode is implanted in the brain and dopamine is released at the neural synapse, it's immediately being measured by the electrode because it's able to measure that fast. So what are we doing at UNLV? We're trying to develop a method for detection of reactive oxygen, reactive nitrogen species. And we are modifying our electrodes with metal nanoparticles to increase our response to hydrogen peroxide. And we'd like to be able to do these measurements in cells. And in the future, once we have our detection platform developed, apply that to certain scenarios associated with cancer growth and tumor development and things like that with microelectrodes. And here's just a representative figure that shows our electrode or our sensor with no peroxide present, and then here's with the peroxide present. So we're actually able to measure a reactive oxygen species like hydrogen peroxide. And again, we'd like to do that in cells in the future. I think I'll skip over this. So to kind of wrap things up, one of the big things we use in my group are diamond electrodes. And you might think of a diamond electrode. Well, how can you use diamonds to do any sort of chemical measurement? Well, if you just dig a diamond out of the earth as a gem and cut it into a gemstone, no, you can't use that as an electrode. But you can actually synthesize diamond in a laboratory with a chemical vapor deposition reactor. And during that process, which I'm not going to describe, boron is added into the process. And boron is one element to the left of carbon on the periodic table, so it has one less electron than carbon. So if you start to put boron atoms into diamond, you create what's called an electron hole, because it has one electron than the carbon that's comprising the diamond. And when you have that electron hole, you're able to shuttle electrons in and out. So we can use it as an electrode to do some sort of reaction. And while there's many advantages, such as a wide potential window, what does that mean? It just means we have a wider potential range that we can do a measurement. So let's say we have an analyte that oxidizes or electrolyzes out here beyond 2 volts. Well, we wouldn't be able to measure that with platinum, gold, or another carbon electrode. We'd have to use diamond for that. So diamond just expands the number of analytes that you can measure because of this wide window that it has. And just so some cool images. So you can kind of see here, this looks like a bunch of little pieces all smashed together, but each one of these is an individual diamond crystal grain. And you know, this is just I think the scale right here says 50 microns. So this right here is smaller than your, does it say 50 microns? Yeah. And so you can see that, yeah, that's smaller than your hair. So you have, I mean, hundreds of thousands, if not millions, of individual little diamond crystals that form what we call a polycrystalline boron-doped diamond electrode. So polycrystalline because there's so many different crystals. And each of these crystals has boron incorporated inside the crystal structure. And therefore, when we have an analyte come in contact with our diamond electrode, and we're applying a potential through the electrode surface, we can cause a chemical change and we can measure the current associated with that and use it as a sensor. So a lot of the projects that we use and what I showed for the reactive oxygen species are using that type of electrode. And furthermore, diamond can be fabricated in a whole bunch of different ways. Here's some little micro electrodes, some like micro needle electrodes. This was used for some brain studies. Here's some flexible electrodes that were also used for brain studies. These were used for some wearable sensors over here. So diamond, because it can withstand this very harsh chemical environment, you can fabricate it into a whole bunch of different ways without losing these really advantageous properties, such as this wide window that you can look at a whole slew of different analytes. On top of that, it's also biocompatible. So you could stick a diamond electrode in your skin and your tissue, and there's literally no response, no foreign body response to it because it's biocompatible and the surface is so inert and non-reactive. How am I doing on time here, Cory? Should I wrap up? I don't know if there's questions.
There's one question. So if you want to wrap up, we can get to the question. And then while he's wrapping up, if anyone has any other questions, if you want to put them in the chat, then we can kind of conclude this out.
Yeah, all right, sounds good. Well, I'll skip over this here then with the nuclear reactors. And I'll just talk about the Phoenix Mars lander and then we'll wrap up. So one last really cool application of electrochemistry is in space exploration. And in 2007, we launched the Phoenix Mars lander that went to Mars to do some soil composition analysis. And here is the Phoenix Mars lander, or like a rendition of it rather. But here over here on the right is an actual image from the Phoenix Mars lander on the Martian surface. And it's collecting some soil over here. And so what does this have to do with electrochemistry? Well, on board the Phoenix Mars lander, there was what was called the wet chemistry laboratory. And inside here, there was little digesters. So the soil could be put into the digester and then it would be, broken down and digested to an aqueous solution. And then it was dispensed into what they call the wet chemistry laboratory. And all it was a series of electrodes that specifically responded to a certain analyte. So we have calcium, magnesium, potassium, some anions, got chloride, copper, cadmium, lead, iron. So all of these inorganic ions can be measured by just simply swapping out for a different type of electrode material. So they were able to do a lot of soil composition analysis via electrochemistry simply by scooping up some of the soil and digesting it on the instrument and putting it into this wet chemical laboratory to do some compositional analysis. So again, hallmark application of electrochemistry, easily miniaturized, portable, and simple to use. So this is a very good application for that. So how do you become an electrochemist? I've said there's some students on the call that might not have chosen a career path yet, but it's very interdisciplinary. I hope I kind of highlighted that today. You know, you can go into chemistry, whether that's physical chemistry, analytical chemistry, chemical engineering, electrical engineering, material science, radiochemistry. I mean, electrochemistry has applications in every single one of these fields, and that's what is what's nice about being an electrochemist. And you know, we pursue a lot of government funding as academic faculty, and being able to work in many different areas is really amenable to a sustained research program. And again, you're just very versatile, so you can adapt as field technology advances. You can certainly adapt, and you know, like I said, 'cause it's just so widely interdisciplinary in general. So in conclusion, I don't think I really have to highlight anything here. Some things I didn't talk about: wearable sensors. Those are really cool. But essentially, you know, electrochemical cells are an essential technology right now, but then also in the future as well. And that's probably largely driven by the dawn of electric vehicles. But you know, electrochemical cells really are essential in so many different areas of life. And if you are interested in any of the, or want to learn more about any of these topics, I didn't really get to talk about the wastewater treatment or the applications of nuclear chemistry. But feel free to contact me. My email's here, my phone's here, and I'd be happy to discuss either of those applications in more detail. So with that, I'd like to thank Cory Nelson for the invitation to speak. My research group members, which are at least about 80% of us, are down here in this picture here. I'd like to thank my collaborators, past and present, from a variety of sources. And lastly, our funding sources, which are all shown here. So with that, I'm happy to take any questions. Sorry I didn't get to show everything, but again, I'm happy to meet, you know, outside of this if anybody's particularly interested in any of those. So yeah, thanks.
Thanks so much, Dr. Rusinek. I think that just gives us an excuse to have you come back to present those two other areas. So we'll have to get you back in here at some point to present again. There was one question in the chat, and it was, can Teslas be powered by solar panels?
Well, they could be charged with a solar panel. It would probably take a pretty, I mean, it's a lot of energy that goes into charging that many lithium-ion battery cells. So in theory, yes. Is that economically feasible at the current moment with the efficiency of solar? Probably not at a large scale, I would say. Maybe if you had one at your house and you had solar panels you could charge your car with, I could see that being a potential application. But with respect to widespread applicability to power or charge Teslas, I think that would probably be I think there's probably still some more advancements that need to be made in the efficiency of solar panels. I'm not an expert in either of those fields, but just knowing the amount of energy that's needed to fully charge a Tesla car is quite a bit, and I suspect you'd need a lot of solar panels for that.
Mm-hmm. Awesome. Well, it looks like there aren't any other questions in the chat, but thank you again so much for your time. Thanks for this presentation.
Okay, sorry I went over a few minutes there.
That's okay. We didn't lose many people actually, so you're doing good. So, cool. As always, thank you again for joining us today, everybody. Thanks for your time. Thanks again to Dr. Rusinek for presenting to us today. And feel free to reach out to him, ask any questions you may have. If you're just dying to know about nuclear energy and how chemistry works in nuclear energy, I'm sure he'd be happy to have that conversation. So thank you again, everybody, and look for the recording to be posted in about a week. Take care.
Thank you.